This computer, got it. So hopefully it will work type thing. So, okay, all right. I think now I don't have a wristwatch here, but okay. Okay, let me get my old wristwatch, because my wall clock was making tick-tock sound, so I got rid of the wall clock. So I think I'm looking. Okay, it's almost 10 o'clock, so I think as people are filtering in, so I have, hopefully I will be able to cover all the material. I think I have. Now, one thing I will want to just advise people that I may skip some slides for lack of time. And so please, at your convenience, look over the PDF, because there is some extra material that I may skip to make sure that I can finish on time. Okay, type of thing. So, and anybody has any questions, comment, advice, suggestions before we get started? Please pick up. I mean, I'm, as my disclaimer is, I'm not a trained teacher, so I will, I'm learning as I go along. Okay, all right. I think, this timer is wrong, but I think that's all right. It's 22 minutes, but I haven't been, we haven't been in meeting for 22 minutes. So, okay, so this is somewhere. Now, this is not my selfie, by the way. It's interesting as to how microbes are in a view. As you can see from this young lady's slide, she's an established investigator named Dr. Lynch. And she did a talk almost November 7th. And you can see from the bottom, it's at UCSF Osher Mini Medical School. So it just shows to the extent of Osher. And it's about an hour and a half talk. And she's primarily an asthma researcher. And I think one of you had a question about atopy and eczema. And her talk is mostly geared toward her. She talks about her work. But it's not, it's a more technical detail. I mean, you think my talk is technical, hers is even more technical. So it's hard to avoid some of the technical jargon in it, but there are varieties of women who are very erudite speakers, depending upon role of microbes. So if anybody has any specific thing, I can send them a link. For example, if you want to know about arthritis or irritable bowel syndrome, there are specific talks related to microbes. So, and if we have, when we have a few minutes, I'll show you my bookmarks about the microbes, so that, and so this is a very, very hot subject in science right now. So obviously, she's much better looking than I am, and this is me. So this is what microbes and me. So, okay, so, all right. And now this is, I think, if I'm not mistaken, I think Robin or somebody had asked me a question about the relationship of food. So I have added some slides. Now, just to revise the material, I'm learning as to gut microbe where. Okay, you can see the picture on the left is it is distributed in this young lady who is doing yoga asana. In the skin, we have relatively small amount. Because the skin is larger, that's why the percentage looks bigger. But saliva is rich on microbes. Stomach has a moderate amount. And dental plaques, obviously. And the biggest stuff is in the colon, 96%. Small intestine is relatively clean. And the concern is very legitimate. We are what we eat. Our moms were right when she said, because lots of this thing is the microbes' new addition. They are our, commensals, as we talked about last time. So, and so this is, just the basic, and the link here are live links. When you see on the slides, hopefully there'll be, so this is like the secret life of skin, because there is more information on eczema and atopic dermatitis on this particular link that I have. I post them on the slides, because if I put them on the notes, they're not active. Okay, and this is basically the impact, just to revise as to what the impact of microbes is. It's like, it's a gut is like a organ with tentacles stretching out in all directions. First of all, in helping us digest our food, to changing our behavior, as you can see from the lower left corner, or synthesizing various vitamins and drugs that we take. And obviously, it's an extra immune organ by itself. And angiogenesis is that fancy word, means new blood vessel formation that is important in cardiology, especially after recovery from heart attack. And also it makes us fat. And then we'll discuss each of this as we go along, that's the plan. Okay, now function of the gut, again, just to emphasize the point, metabolism, SCFA, this abbreviation means small chain fatty acids. So as fat that we eat breaks down into fatty acids, and that's how the nutrition is metabolized in our liver primarily. Now we cannot synthesize these two essential vitamins, and so this is done by microbes in your gut. And again, the drugs that we take are metabolized and in the liver and excreted in the kidneys. And then again, salvage calories, because what you eat, obviously has to be broken down, then the body can utilize it into smaller molecules instead of complex molecules that we consume. And again, the bile acids help us digest, and that's liver is the big powerhouse. And prevention of bad pathogens, so it's like, and as we talked about in the first two, like they can go from good to bad and from bad to, so there's a nice harmony going on. And it's the immune organ, as we talked about, and we'll dwell more into it as we go along. And this is just a cartoon to show how we can go from disease to health and health to disease and these are the varieties of abbreviations that we'll talk about. I already explained this small chain fatty acids, antioxidants, they were in the news for the last several years. Insulin, you already know about. And getting on to the right, it talks about lipoprotein polysaccharides, which is simple sugars. CVD stands for cardiovascular disease. And TMAOs, we'll talk about, that comes from meat that we consume. And so the red are the bad guys, and the blue are the good guys, so to speak. So, and the harmony goes between our food that we take from sugar diet on the top, and it's basic chemistry involved with pH, and the probiotic that sometimes we take, such as yogurt. Okay, and these are again, just to say who we talked about in the first two sessions, the good guys, the bifidobacteria that you get in the breast milk, the E. coli, there are the largest proportion in your own lactobacilli, which are again in the milk and dairy products. And the bad guys are at the bottom, as you can see, the ones that cause diarrhea. So we talked about what, and then who, and then how. Okay, now this is, I think we talked about symbiosis in biofilms. I just found this recently type of thing. This is hopefully this will.
Biofilms are essentially groups of bacterial cells, communities that are encapsulated in a matrix that the cells themselves produce.
They can actually secrete something that will modify their environment. It's something for them to stick onto, such as a rock in a stream, or to stick onto your teeth, and that's where we get plaque from. What we're trying to do is to take a naturally occurring evolutionary technology that bacteria use to create their own structures so that they can survive, and actually repurpose that for our own ends.
The concept of the microbial factory has been out there for a while, but what we're really trying to do that takes it a step further is not only use the microbe as a factory for the production of raw building blocks, but also program that cell to orchestrate the assembly of those building blocks into higher order structures and then maintain that material over time.
We call our technology BIND, which stands for Biofilm Integrated Nanofiber Display.
And what that essentially means is that we have programmed bacteria to be able to produce living materials in the form of bacterial biofilms.
And the way it works is we genetically program the bacterium to produce artificial protein. This protein is a fusion protein composed of an amyloid domain fused to a functional peptide domain. And we can mix and match that peptide for whatever end function we want.
The cells produce this protein within themselves and then secrete it through various biosynthetic machinery components into the extracellular space. It then is nucleated at the cell surface to form something called amyloid fibers, which are essentially these self-assembled protein structures that form fibrils.
These proteins will stack on top of each other, much like Lego blocks will fit into one another, and they'll keep on growing until we actually generate fibers that are microns in length.
The fibrils eventually grow large enough to encapsulate the cells in kind of a meshwork. And this meshwork is the biofilm material that we are engineering. What we show in our paper is that this basic mechanism of...
Okay, so this is about as cutting edge technology, as you can see. There is a giant of so-called new field called synthetic biology at Harvard. He's a man from Florida. His name is George Church. And this is about as cutting edge technology, as you can see, about biofilms, which we talked about in the teeth, or the dentist always do that when you go for the dental cleaning. And this is not just in your mouth, but it's also in your intestine. The bacteria they are very collaborative creatures. They pile on top of each other and form a nice layer underneath, on the other side between the mucus. And so this is, and that just gives us the protective layers and type of thing. And when there is breach in this, and this may even have a role in future treatment for antibiotic resistance that we talked about. And this was in the news, the picture, I don't know if somebody saw this in the email that I sent out. This is a picture of a chimp, and now he has this bumps that you see. And this is the first time, this is about as hot off the press as you can see. We were talking about in the earlier thing about leprosy. This is a chimp this is the first time that leprosy has been diagnosed in other animals other than humans. So this is chimp with leprosy that made the science news recently, just to show as to how the old disease hasn't totally disappeared. So, but
biofilms.
Okay, and this is a new thing about, we talked a little bit about quorum sensing, and again, antibiotic resistance has been in the news. Just the other day, Financial Times from UK had an 8-minute video clip on antibiotic resistance, which may increase with COVID because people are using antibiotics, just to prevent pneumonia. So even, we are hitting them with everything we got. So when we look about hundreds of thousands of people getting multiple complex antibiotic, that is, again, we are, seeding a germ seed for future antibiotic resistance. And this is an interesting thing about people are using something called quorum sensing. Bacteria like to live in groups and colonies. And these are and they talk to each other. Well, intraspecies means within the same species, and same layer, the interspecies with different type of, So what this particular group of researchers named Bonnie Bresser did was they devised small molecules which can block this communication. You can see the round and triangles outside the circle are the tiny molecules that they have synthesized, which block this crosstalk within the same species. For example, like Pseudomonas or E. coli or Vibrio, which is called, type of cholera. And interspecies means one species talking to other one. And so, and now by bacteria, when they get to a certain number, they can cause an infection and causing the animal to die. But when they block these particular interspecies talk by the small molecule, the animal stayed alive. So this is a new way of, this is like the bacterial Esperanto that is being blocked by small synthesized chemicals. So this is one way of going our antibiotic crisis that we are we are already facing. We have ignored the problem, but so this is one cutting edge technology that is being researched upon. Okay, now again, I've added some more slides for food, and the two essential foods that we eat and that are useful are tryptophan, which is metabolized into various chemicals, as you recognize the serotonin, which has a cause for depression, and antidepressant drug called SSRI, serotonin blockers, and that converts eventually into melatonin, which helps us sleep. And so this is, and tryptophan, again, Thanksgiving is coming up. It's rich in turkey and other foods such as eggs, cheese, and also other chemical that we'll get to, which is, let's see, which is called tyrosine, that is widely prevalent in almonds, lentils, seeds, edamame. And the third chemical that is important is called ILA, indole lactic acid, which is metabolized into propionic acid. And this leads into something which we all know as called antioxidant and free radicals. So these are all the foods that bacteria help us digest. And so this is, a part of the foods that are. Now, where do these foods come from? Again, from our diet, the meats and tubers. And again, so it depends upon how the food is processed. The cooking has a big value, because by cooking, we make it more simple to digest type of thing. And that changes the microbial flora. And again, for example, like some of the chimpanzees, they have a large gut because it takes them a lot of time to digest the food. So that's why they spend, almost 12 hours eating food. While by cooking and evolutionary, as human beings have improved our calorie balance by consuming relatively small amount of food, we can get all the necessary calories and leading to enlargement of our brain. Because otherwise, I may have mentioned, but it's worth repeating that because brain has such a high calorie requirement that one of the scientists did a calculation that if we actually just had uncooked food, we'd have to eat almost for 24 hours to provide enough calories to supply enough calories to our brain metabolism type of thing. So this is one of the big advantages for evolutionary has occurred type of thing. So, and again, the microbial flora changes based on diet. People did interesting studies on immigrants from Thailand to US that their microbial flora changed in as quickly as within 9 months from moving to industrialized society. And again, this was not necessarily bad, but just different as to how diets affected. And similarly, people have done research in elite runners that they have different microbiomes than non-runners, so to speak. So there's varieties of interplay that takes place between various communities depending upon their lifestyle and the diversity of your diet. So the important part is not just what you eat, but how much diversity you have in different food groups that you're talking about. And people have gone into really closest to hunter-gatherer cultures in Africa. These are the people called Hadza, I may be mispronouncing it, Hadza. And they eat as much as 150 grams of fiber per day, which is 10 times more than the average US citizen, US person. Okay. And again, people compared our eating lifestyle with, and we're not, evolutionary, we're not closely similar to chimpanzees, but we are more like bonobos. These were the apes that were living in the plains, not just living in the trees and eating fruits. So this is our lifestyle is more like bonobos. So the important point that needs to be emphasized, the intake of fiber and the diversity as far as food is concerned. And this is the lactic acid and the propionic acid, that is the antioxidant and free radicals that we talked about. And this is the molecule called tyrosine. And the tyrosine, which is again metabolized into the chemical that you recognize as dopamine and epinephrine. So these are the important elements that we get from our food. So this is enough about food and the type of nutrition that we get. So last but not the least, this is what the cartoon just shows us. Every 35 days, we are replacing our entire skin and various new cells. So So we are actually not you because you're being replaced all the time in terms of human metabolism. So the food is extremely important in terms of how it interacts with our microbiome. Okay, and again, we just to answer the questions, we talked about atopic dermatitis last time, and I think I didn't do enough justice to that, so I put that slide again because the eczema and atopic dermatitis is increasing. It's about a threefold increase in 30 years, type of thing. And this may be, again, due to changes in our microbiome. And various companies are migrating. There is some company which is making a cream called Pacifica and various probiotics that are being marketed. But again, those are to be taken with a grain of salt because they haven't been well established for current use. And at times, the FDA has been, some of the claims that were being made for some of the probiotics were rather tall and they had to notch down their sales pitch. Okay, moving down from front onward, starting with the nose, we talked briefly about it. And again, the Staph aureus is in our nose. But again, there is enough mucus level that you don't get infection. So our body has a way of protecting it. And when that layer breaks, that's when you get the serious sinus infection that can go to your brain and can cause some serious problems. And again, the microbial flora has a lot to do with when it is disturbed and dysbiosis takes place, it's causing rhinitis and again, bacterial infection as well. And some companies have tried to migrate nasal probiotic, but again, that's a little more hype than real proof type of thing. Okay, and okay, this was the research from the first slide that I showed Dr. Lynch, and this is her work. And now I couldn't resist putting this. This is the Oxford Dictionary search for the word asthma. As you can see from how much the word has become more common frame from 1960 onwards, it just shows how the increasing asthma that has taken place from 50 onwards. And that may be more with better public hygiene and change in our microbial flora. So even just the word search in the dictionary and the word usage has increased. And now what Dr. Lynch did was they took newborns and they isolated their meconium, which is their poop, by the way, into 3 separate flora. And the 3 color-coded from blue, green, and red. And as you could show that different, depending upon the microbial flora, right from the newborn age, see that the incidence of asthma was different. As you can see from the rather complex chart, the incidence was, they could predict right from birth onwards as to which baby was going to get asthma and atopy based on their microbial flora. And as to see in this chart that's on the left bottom side as to who had more diversity type of thing. So this is how they were able to classify depending upon the microbial flora in the poop of the newborn baby.
Right.
Yes, please, go ahead.
I think in the 1950s, they weren't diagnosing asthma very much.
Yeah.
I know I grew up with with bronchitis, but it was caused by allergic reactions.
Mm-hmm.
I was never called asthmatic till much later.
Yeah.
I think that's not uncommon.
Yeah, but I mean, we're talking about right now, we're talking about, so in 30 years, so from 70 onwards, so there is definitely data that in the last 30 years, we are already in 2000. So after 70 to 80s, numbers have definitely increased dramatically. So that the data is there elsewhere type of thing. So this is, this is a word search type of thing, but the incidence has clearly increased over the last, now, I don't know whether it has increased from the 70s or 60s type of thing. So again, obviously medical diagnosis dramatically has changed, from the 70s and 80s onwards type of thing with the availability of more specialists type of thing. Because as we talked about in the history of medicine class, again, there are a lot more pediatricians, so they're more tuned perhaps in picking up the diagnosis as opposed to the family doctor type of thing. So I mean, that's, but again, so I entered practice like in the the mid or late 70s. So your probably experience is deeper earlier than mine. Nice. Okay. All right. Okay. So we talked a little bit about the diversity in the newborn babies, and the gut microbes actually. So one can say that it kind of flips the immune switch and to various allergens, and that may be again related to hygiene hypothesis that we alluded to earlier. And again, the flora is different, and the two words that we had emphasized called bacteroides and proventales. So, and we'll talk more about it when we get down to the gut level. Okay, and so again, the gut microbe manipulation. Now, a lot of the research is done in mice because they are the good model for this, and by changing what Dr. Lynch and others people have done was to take the microbial flora from the humans with affected and have infected so-called germ-free mice and seeing if they can get the airway mucosal response. And she has some, for lack of details, I didn't put some of her slide, but they showed when they infected the mice, the so-called new germ-free mice with the poop from people with disease, that their airways definitely constricted, and they developed, on pathological slides, asthma-like picture. Okay, and again, this was, and their T cell responses changed. And so this was, her work is really elegant as to how they proved the whole hypothesis by confirming various anatomical models and experimental and lab work type. Okay, now getting down to the mouth, okay, now
Right, can I ask a question? On the previous slide, could you remind me again what the difference was between 1, 2, and 3?
Okay. Oh, in terms of the diversity of the microbial flora, so this is, like the so-called, pie chart that is there. So, it's like the.
Yeah. But the infants,
yeah.
It was just what they measured. They didn't treat the infants differently.
No. They just,
okay.
It was just, see, based on metagenomics, like what they did was they took the poop and they did the, 16S diagnosis of the microbial flora and compared with the large scale data, because they couldn't grow things, because the only thing that would grow would be the most prominent bacteria, which is E. coli, but the smaller ones wouldn't grow in the petri dish. So what they did was the data analysis, based on this ribosomal analysis, compared the data, basically.
I get it. Thank you.
Stuff would not grow.
Yep.
And obviously they didn't do any treatment, because it was, there'll be, we don't know what we are treating, so to speak, so, and would create problems. So, okay, and again, when we get down to the microbes, so I think when we showed the site of the Microbiome Museum in Holland that we talked about in class 1, as to how many germs you exchange when you kiss somebody. And so mouth is just like absolutely a big ghetto of microbes, so to speak. That's to put the word, There are about anywhere from 300 to 500 different species type of thing. And that goes back to the classical thing that whenever you went to a good old family practitioner, they always looked at the mouth, as the tongue was the window to your health. And again, there was like a differential diagnosis as to how many diseases can you diagnose by looking at somebody's tongue. That goes back to the ancient eras from the Chinese and the Indian and the Greek medicine days, before the advance of modern medicine. And again, the plaques and other things are well known, which the dentists have been fighting for a number of years. And again, these are, when with aging, the saliva decreases and the biofilm that we talked about, that the first slide, the Harvard guys talked about, has a huge value. And it's based off a complex molecule called polysaccharides. And this, again, in which there are the bacteria, they're lying on top of each other, like they're building up literally a skyscraper. They are one colonies on top, some cells that are right next to your epithelial cell, and the others are sitting on top of it. So it's almost like it's like a real estate, the location. So as to how the bacteria kind of colonize and form the quorum sensing as to they keep the company. And so the mouth is absolutely a big powerhouse of microbial flora, and it's affected by Now, again, because you have to realize that a lot of the bacteria in aerobic or mouth is filled with oxygen. So they have to make they select their location very carefully. And again, so some of them are sitting right next to taste buds. And again, it's a really interesting things. And you change it every time you use mouthwash, you're changing the flora dramatically. Okay. Now, it was interesting to find that when you go for teeth cleaning, how long does the effect last? The dentist go through all sorts of things and scrape and work on it for hours on. How long does it last for for bacteria to come back again? Within a matter of hours. So the dentists have done an excellent job of, almost making mouth clean and healthy, but the bacteria just, claim the real estate very quickly. So it just goes to show as to how powerful their role is.
So, Bharat, what do you think the balance is there? I mean, it is important that we get our teeth cleaned, right? Are we getting rid of some
Yes,
things?
yes. Oh, okay, yeah, because you're getting rid of the pathogenic this thing type of thing. But again, there are some of the primitive communities, they've never had this thing. So somehow they've developed enough immunity type of thing that they've, but yes, I mean, I'm not saying, because obviously as cardiologists, I think yesterday you were asking about the question about heart valve infection and in Dr. Spector's class. And while the rheumatic heart valve disease has gone down, one of the most common reasons for heart valve disease now is endocarditis or heart valve infection. And for that, the big reason is that we talked about as to the oral sepsis, from your mouth, the bacteria can go and affect the disease valve. So that's one of the common reasons for causing endocarditis. And it's a pretty serious illness type of thing. And that it requires, a substantial number of heart valve surgeries are due to infections, especially in people with substance abuse, and even in just people who have had just unlucky that the bacteria managed to break the barrier and go to your bloodstream and cause a bad infection of a deformed valve. Okay. And this was an interesting, I couldn't resist this picture, that this is a knockout picture of somebody actually grew the colony, and this is like the image as to, and they managed to stain it with a different way as to how the epithelial tissue is gray in the middle, and the various colors indicate different bacteria as to some are on the outside, some are on the bottom. So it's make for an interesting, as to people have a lot more time and a lot more technology as to how the flora can, you can see it's like it's a variety of almost like that New York City, in the 1930s, all sorts of population is living together and diversity that is there. And a change in any of the diet, saliva, and hormones leads to dysbiosis and systemic diseases. And again, this is, the classical example of dental caries. The various factors can affect, starting from stress to environmental changes, to ecology, and leading to disease. So this is just the so-called plaque hypothesis. And increased carbohydrate consumption can disrupt the homeostasis between good microbiome and dental health. So it makes sure that we are providing them a rich culture medium. And we already talked about how the dental cleaning will change very quickly. And this hypothesis that teeth can be a cause of disease is not relatively new. It was proposed as far back as 1900 by some physicians, and rheumatoid arthritis was supposed to be related to, teeth infection. But then after a while, they thought kind of fell out of favor, only to be reinvented later on in the 90s with the better culture technique and better rise of genomics. Okay. And again, and we'll talk more about the arthritis things, as to varieties of proteins are, the one important protein called citrulline is affected by the bacteria in your mouth, a very fancy name, which is of academic interest type of thing. And so there is a role for good dental care, not especially in type of arthritis. And there's a whole 1-hour class on it, that one of the ladies, did from Wisconsin on the role of microbiome in various types of arthritis. and the role interplay of immunity, microbe, and various degenerative and rheumatoid arthritis. And again, obviously, not to underplay the importance of smoking that changes the diversity in your mouth. So it not just affects your lungs, but it also in your oral cavity. It will change the microbial pattern because again, you're putting smoke, which is bacteria are not happy with getting exposure to carbon monoxide and nicotine. And now this is, just like the survey of the numbers that we said. So we may have talked about it earlier, but it's worthwhile going over again. Stomach, 10 to the power 2. This is logarithmic scale. Duodenum, again, not that rich. The first part of the intestine. The jejunum, which is further downstream. As you can see on the left, the small intestine is relatively paucity of microbes. And the ones that are there are so-called good guys, the lactobacillus that helps you digest, yogurt and the benign type of, obviously, we'll talk a lot more about the Helicobacter. That's the big poster child of microbial flora that's in the stomach. And when we get down to the large intestine, you can see we're getting exponential rise of 10 to the power of 12. So that's like, you can say, well, there are more bacteria and viruses than the number of stars that are in the galaxy and the universe, basically. So it's a huge number. And the primary flora is Bacteroides, Enterobacter, E. coli, and so the varieties. And sometimes some of them can become pathogenic very quickly. Okay. All right. And now, again, this is the dark side of microbiome. So these are the pathogenic type of things as to what the diseases that can be created. The one that is obviously Helicobacter, that's related to ulcer disease. The important one is inflammatory bowel disease, IBD. That is, the two primary diseases are so-called Crohn's disease and ulcerative colitis. We already talked about allergies and asthma and rheumatoid arthritis. They have a role in, colon cancer especially. And there's a lot of research being done on relationship of gut microbes to some of the neurological conditions. The big one being Parkinsonism. There are some thoughts about autism, and even in diseases such as schizophrenia, there have been, and behavioral disorders, it's been implicated. And the metabolic diseases are a big factor, a relationship of abnormal microbial flora. And the famous one is, we talked about, some of you were in the medical history class, was the H. pylori. And that was back in the 50s, we always thought the ulcer disease was related to stress and anxiety in our lifestyle. But again, the Australian pioneer who did the paradigm shift, and he saw the bacteria, he was working with a pathologist and did experiment on himself and drank the culture of the bacteria, and definitely acid rip-roaring ulcer disease. And again, they were rewarded with the Nobel Prize for their groundbreaking discovery on the role of gastritis, aseptic disease, and relationship.
Rita has a comment.
Yes, please, go ahead. Please unmute yourself.
Unmute.
Hello.
While she's unmuting, this is really a deserved Nobel Prize because it made a difference in people's lives. Because we treated the disease how it's supposed to be treated.
So yes,
we treated the cause, and it's just magnificent.
Yeah. And now,
Rita, if you go to the bottom of your screen, to the left, you'll see where to unmute. You see that? Somehow I can't unmute her.
Yeah, I think I don't want to go off my slides, because
No, I'm on there. I just can't unmute. Rita, can you hear me? If you go to the left bottom of your screen with your pointer, with your little arrow, you'll see where to unmute.
So, okay, yeah, sorry about that. Yeah, apologize. And so this was, the ground, but there are some other contrarian thoughts on it, from Dr. Blazer type of thing that, yes, that now we're, at least in the West, in the Western society, we said that only good H. pylori is the dead H. pylori, but it's been around human beings for ages. And maybe it wasn't all evil as it was thought, that perhaps by eliminating H. pylori, that we may have caused more reflux disease and GERD, as we talked about. And Dr. Blazer, who is one of the pioneers in microbial research, and at the end I have the book called Missing Microbes, and he makes a point that perhaps we are overdoing it on eliminating H. pylori totally. And not all pylori needs to be kept. So again, so there may be some evolutionary advantage to that. For example, so not all bacteria are necessarily, they've been around for ages, that, and they think, now I have to double check on my reference, that Otzi, the Iceman, they were able to isolate some of the signature of some of these bacteria going back to thousands of years. Okay, so his point is that perhaps that we might be treating it over enthusiastically than we perhaps needed to, and that's a different viewpoint type of things. Okay, and his book called Missing Microbes is definitely worth reading if you are looking to drill more into this subject. And I have several references at the end of the talk. Okay, and we already talked about the breast milk benefit earlier. So, and this is again, the micrograph of H. pylori on the surface of intestine, just for, just to show as to what that bacteria looks like, and like a corkscrew type appearance, and just the technology that is there. And again, the layer of mucus is pale mucus that gives us the protective layer. It makes a peace between the bacteria and your blood and your circulation and the lymphatics behind. So it kind of makes it, okay, keep the two warring parties separate, so to speak. And again, so it forms a nice layer type of thing, okay? And the varieties of other organisms are also related. For example, the so-called methanogens, which are part of the normal flora, which, makes you fart, okay? And again, there are other organisms which help you digest, the lactobacilli that help you digest the other. Okay, now this is these are the two common flora that we talked about, the two important words that are like to be remember. It's called Bacteroides and Firmicutes, and these are the two phyla that are prominent. This is what they look like on picture, and Bacteroides and Firmicutes, so, and this is the appearance of that, so like a small, organism, not a rod-shaped organisms that are there, and they stain differently on microscopy. Okay, now this is an interesting tidbit type of thing, autobrewery syndrome, type of thing. This is some guy who got drunk by eating fries or carbohydrates. And this is the condition, that's just of curiosity, so to speak, a rare condition called autobrewery syndrome. The bacteria that are inside their gut, which changes into alcohol. Okay, so this is endogenous alcohol. So he was, he had enough blood levels to be classified as intoxicated, while he was totally denying he didn't have a drop of alcohol by mouth. But it was his bacteria that the high sugar and high carbohydrate diets, led to endogenous fermentation by the fungi and inside his gut. And so again, so this was involved in, and he was able to change it by changing his diet. And again, the poop to the rescue, so fecal implant helped him, get over the condition. So just out of curiosity, and I'm sorry.
Brett, I was going to say, I think last Wednesday, the show Chicago Med, they featured a patient who had this disease. Of course, it was all this drama around that she came in drunk, but oh, I hadn't had anything to drink, and it was exactly this. So it's funny.
Yeah, so it's an interesting thing. I mean, it's very uncommon, but just, curiosity type thing, it gives you, provides a good diversion, makes a good news story. Okay. Again, but more importantly, it's that some of these bacterial production may have been involved in development of non-alcoholic fatty liver disease, which is a lot more serious condition, okay, than this, because it left unchecked, that's a it's not an uncommon disease, that's not all liver disease is related to alcohol or hepatitis virus. Okay. Now this is again, so this is from the slide is from Michigan Medical School, the exposure up to C. difficile. So difficile.
Here's your shirt.
I'm
Where's-
sorry. Okay. But moving on.
Show the money.
I'm gonna try not to
We need to get the bills based on what shows.
I have to mute. Okay, I got it.
Okay, should I stop or, okay, oh, I think she was just muting herself. Okay, now, the other important things that makes the gut microbe in the popular press and makes a huge story was the fecal implant type of thing. And this was the disease which is again called C. difficile, which is due to a large, exposure to a broad spectrum antibiotic for several days. And it's a very serious life-threatening illness. And this is the pathological slide pictures that this is after exposure for 24 hours, 30 hours, and you can see how the inflammation takes place from the toxin that is released by the commensal fluoride, which turns into pathological and overgrows and causes, a huge diarrhea and serious illness that you could almost, by the metabolic damage that can take place, people could actually die from it untreated. Okay. And again, initially the treatment was with more and more strong, powerful antibiotics, and the response rate was at the most 50%. Okay, and this is where now the fecal implant is not a new thing. It has been reported in the Chinese medicine and Indian medicine for a long time. But in the Western medicine, the first trial that was done was in 1958. But the first control study was done sometime in 2013 for C. difficile infection. And the results were so dramatic that they actually had to stop the study before the prescribed time, because they just thought it was unethical to continue, because the people who were analyzing the data showed that the people who had the fecal microbial transplant had a dramatic success of 90% versus 50% in the conventionally treated people type of thing. So, and that just, again, this was like a groundbreaking report done sometime in 2013. And again, now, it's in spite of that, this does not give us a license that we should all try this for all sorts of illnesses. And again, the indications have been expanded dramatically, but doesn't mean you should do home treatment therapy by saving your poop and doing transplant for all sorts of illnesses. For example, if you're a child with autism, do not try this at home, or a grandchild, okay? Because there is a risk. And most recently, this is I just came across within the last few days, the first death was reported with fecal. This is medically supervised fecal transplant in 2019. That this was somewhere at, I think it was a general, that the report was that they had an unusual strain of E. coli that caused severe overwhelming sepsis, and the person died from it. So this was the first report of a dangerous outcome, or fatal outcome, in 2019. So this has been, and again, this has to be properly screened to make sure that you're not putting any pathogenic organisms. And there's almost people who are so-called super donors have been, it's like a blood group, for example, the O negative is considered, the super donor because it is so rare. The same way people who have never had antibiotic exposure or whose stool has been analyzed to make sure there are no pathogenic organisms in it, And so they maintain a list of so-called super donors. Okay, let me make sure. Okay. Oh yeah, I'm recording. Sorry about that. Yeah. So this is, the big dramatic change, especially in this particular infection. And this is how it takes place. Again, just to give you the more scientific basis of it. Let's start from the top, from the normal. It's more diversity that you can work your way down from top. The bile acids, the synthesizing microbes that break down from primary to secondary bile acids. And again, that interferes the arrows that goes down from the extreme left, the bile acids, they break down the C. difficile spore and into a difficile spore. And again, that prevents the one arrow with the flat line around here shows that the growth is inhibited by the secondary bile acids, and that prevents the toxin production. Okay, as you can see from the left side, hopefully I can see my arrow that in charts, the flat arrow is, so it's an inhibitory effect versus working as adjunct effect. And while I've got on the right side of the picture, the antibiotics perturb the flora of the bile acid. The conversion does not take place between so-called primary to secondary bile acids, and you lose the inhibitory effect, leading to toxin production from the C. difficile. And that, again, the chain reactions start with increased toxin production and inflammation, causing the disease. So this is, as you can see, the liver and the bile acids have an important role to play in causing problems with C. difficile infection. And again, so this again falls right into the category that the liver plays a dramatic role, as we can see, start on the slide, as to increase endotoxin production, causes leak in the epithelial layer, okay, and releases the inflammatory chemicals, the small molecules called cytokines, and again changes the flora, and again it leads to problems in the livers, what is typically called as portal systemic, the circulation inside the liver, and changes into the systemic shunting of blood flow, causing the toxins to go into and causing liver disease. Okay, so this is again, so the gut microbes have a lot to do with it. Now, just to give the ABC, some of you have taken the immunity class, but just to go over basic immunity, immune system is wide, spread wide over in the body, starting from the tonsils to lymph nodes to the biggest organ is spleen and the Peyer's patches in your intestine. So this is where the immune system is related. And the two types of immunity that is well known, especially nowadays, the knowledge of immunity has increased dramatically. The so-called cell-mediated, so the T cell and the humoral antigen antibody. And this is the classical antibody formation with the heavy chain and the light chain. That, so that's just the basic immunology 101 that's been, I'm sure that people are becoming very knowledgeable about immunity in the current crisis. Okay, and we've talked about when you have combined immune deficiency, the so-called David Vetter, who was the poster child, and the bubble boy in the 70s, and it was the classic example. And the various immune globulins, which we talked about, the so-called IgA, which is related to influenza, related to a nose, ear, and IgG, which is in the body fluid, can cross the placenta, IgM, that's a response to infection, IgE, that is related to pollen, allergy, fungus. And again, the immune system does not develop right away. And this is, I'm just summarizing all the details, and so when you're born, it takes a while to develop for several months to develop, and you're dependent upon your moms. And so it shows that, so this is the role, just the basic of it, just to bring you up to date on the role of immunity type of thing. Okay. And microbial guides now, again, the microbes have a lot to do with the development of your T cells. And you can see this is the naive T cell and the microbial flora, and there's cell called T cell helper 17 causes inflammation, while the Treg cell it suppresses the inflammation. So again, not to get bogged up into the real nitty-gritty of immunology, but this is, again, if you can just say, for example, the T cells have a complex interplay with your microbial flora in causing inflammation to suppressing inflammation. Okay, so this is, this is all we need to know for our class, basically, type of thing. And again, the inflammation has a huge role to play in Crohn's disease and again, various neurological conditions as well. Okay, all right, moving right along. And this is, again, this is even more complex type of thing, as if you didn't have. So microbes, again, have a role with various immunomodulating and various chemicals that we alluded to earlier called small chain fatty acids, again, and the various vitamins. This explains the role of pro-inflammatory on the left side and anti-inflammatory on the right side. And these are the complex immune chemicals called interleukin. The TNF stands for tumor necrosis factor. And so this, you can just see how the inflammation is a rather complex interplay that takes place between the breach in your intestinal mucosal barrier that takes place, as to how the crosstalk takes place between the microbes and your T cell response. So if there is a break in the mucosal barrier, that's when all the hell break loose, so to speak. And again, the knowledge of that is extremely complicated. And again, so this is varieties of called gut-associated lymphoid tissue is called GALT, and mucosa-associated lymphoid tissue is called MALT. So these are the two important organs that are part of our immune system. Okay.
Brett, can I ask a question? Were you implying on the previous slide, the one before this, that microbes are important for, like, we need them to do some things, but then they can get out of whack? Is that the idea? Or
Yes, oh, absolutely, yeah. So it's a treating.
We need them to make that transition from the naive T cell to the Th17.
Yeah, so yeah, you definitely need them type of thing to, because it's like, you need the good microbes there type of thing. So this is, again, obviously, and when the flora changes, that's when the inflammatory process takes place. So I have a few more slides about the inflammatory bowel disease that is coming up, type of thing in a few seconds. We're getting to that.
Thank you.
Okay. And so again, so this is the complex interplay that we talked about in, okay. And so this is so-called immune. So you're not just getting old, and as we get older, our immune system changes, type of thing. And so the gut microbiota has a lot more role to play in aging process as well, type of thing, because our immune system response goes down after age 25, and the inflammatory processes increase, type of thing. That's because with aging, you can just see how the inflammation and the epigenetics is the genetic code that is not, there's no alteration in the DNA or RNA, that thing, but the simple molecule called methyl or acetyl molecule that changes, and it changes. So the importance of environment on causing inflammation. Again, it's like we talked a lot more about stress has a role to play, and again, metabolism, stress, diet, and again, I think I talked a lot more about in epigenetics in the so-called the starvation that took place amongst the Dutch during Second World War, where the people were starving and living on like less than 1,000 calories, and when the food became available, not only the next generation became obese, but even the third generation became obese. And that had a lot to do with your epigenetic modification. That's the classic example of as to how environment affected the outcome and changed the whole interplay between the microbial flora, their genetic messaging system, and that took place even skipping a generation apart. So that was a classic example in the science of epigenetics, that took place. So this is, again, so as to how varieties of factors come into interplay and in causing inflammatory diseases. And so the crosstalk that takes place with various organ systems. And so the simple thing that we are talked about, simple one molecule change called methyl group changes. So, okay, all right, okay. And the varieties of autoimmune diseases have a huge role to play, as to see now, as I think 2 or 3 of us were in the immunology class yesterday, and I think 4 of us at least that I can recall. And these are the varieties of autoimmune diseases, starting from the top onwards, the prime example being multiple sclerosis, okay, and then the lung disease called Goodpasture syndrome, and obviously type 1 diabetes, glomerulonephritis, rheumatoid arthritis, and again, the inflammatory bowel diseases, So these are some of the, and the varieties of factors that play that we talked about, called genetic predisposition, your hormonal changes, again, the autoimmune disease, that's one of the reasons why they're more common in women, microbial flora, and the role of so-called molecular mimicry, and the HLA, the human leukocyte antigens. All of these factors play a role in leading to inflammation. Okay, so, and this goes to show you this is evidence. For example, people did studies between 2 close areas of the relatively more affluent area of Finland, versus the nearby area, nearby Russia, an area of Russia called Karelia. Okay, and since 1950s, the incidence was, there was a dramatic difference between the 2 areas of Finland versus Russia. There was like a 10-fold change on 2 sides of the region. Okay, so a 1/6 type of thing, in nearby Russia type of thing. So the incidence of diabetes was that dramatically different, between the affluent area versus the more traditional area of Russia. And the similarized we have seen in the United States, and the same way the problem was, Yeah, the same problem was in asthma, and we've seen similar areas and we already talked about eczema type of thing. And this has, again, may have a role to play with the so-called hygiene, and maybe we're becoming too clean. And a Canadian researcher, Dr. Finlay, has a book as well as a talk at the Royal Institution of so-called "Let Them Eat Dirt." And so that's as a huge example as to how preventing the diversity of microbiome may have a big role in the rise of autoimmune diseases that we, Okay, I think, did I miss a slide? Wait a minute, let me go back. Okay, yeah, here we are. Okay. Yep, I think, yeah, okay. All right, and so, and we talked about that some of the diseases are more common in women, and that may have a role to do with hormones, with estrogen and progesterone type of thing. Okay. And now inflammatory bowel disease, again, the imbalance between the two types of T cells, the T helper and T regulatory cell, that takes place, and that may be driven by changing your microbial flora, leading to change in your inflammatory reaction. And again, how we cannot underemphasize the importance of mucosa, and that breaks the barrier and it changes the layers of the mucosa, and leading to the 2 diseases that are the prime examples of inflammatory bowel disease, the Crohn's disease on the top and ulcerative colitis. And the treatment approach is different for both of these diseases type of thing. And the new anti-inflammatory drugs, which are the biological drugs that have made a dramatic impact. And I think the one that is overly advertised on television called Humira, and the brand name is called Infliximab, and that has played a huge role in preventing the a flare-up of Crohn's disease, because originally this was treated with antibiotics, and the one of the earlier drugs that you may recall that was struck was sulfadiazine, was used for treating Crohn's disease and ulcerative colitis, but the response rates were rather poor to begin. But the new biological agents have made a dramatic difference, and obviously, they're expensive drugs, but they've prevented a lot of patients from needing extensive colon surgery, because the treatment that when we were in medical school was, if there is a lot of inflammation, and that was to remove large part of colon, with leading to various problems of short gut, and eventually some of the patients, and there was also a risk that some of the patients eventually ended up with cancer, left poorly treated. So as you can see from the slide, it forms granuloma, which is the so-called benign tumor, and subsequently leads to malignancy as well. And there are several types of this. There are two forms, the one with the so-called constipated type of problems, another with the diarrheal or mucosal diarrhea type of problems. So there are various presentations that takes place of the inflammatory bowel disease. Okay, and again, this is just puts into the same thing, better cartoon effect as to how the homeostasis is disturbed by cytokines in between the Treg and T helper cells. So again, just beleaguers the point as to how the inflammation plays a huge role. Now, moving on to the more common problem, which is obesity and metabolic syndromes, because over the last 25 years, in spite of varieties of diets and spending billions of dollars, the number of obesity has increased. And again, the evidence may be that the change in the microbial flora as to how we are getting calories from our food and storing may have a role to play, that we may have been underestimating it, that it wasn't as simple as calorie in and calorie out, that the change in our microbial flora may have a huge role in causing, because we all know people who said who can eat anything and don't gain weight. At the same time, people who have followed diet meticulously and not able to lose weight. And if anything, they gain weight, so to speak. So even with diet, and only to get a poor response. And this may have a role with the so-called balance between our firmicutes and Bacteroides. And there are some bacteria that actually can cause weight loss. There is one particular species that has gotten a lot of publicity called Akkermansia. And that has been known to help with weight loss. But again, the state of the art is not such that you can get a probiotic of Akkermansia and help you lose weight. So because a lot of the research is done in mice, and again, that does not translate very well into a human being. And the interesting research that was done was in two illnesses that occurs, not so much in the Western world, but in sub-Saharan Africa. And it was not uncommon when I was in medical school in India, the two illnesses that you may have not have ever heard about now was called marasmus and kwashiorkor. Kwashiorkor is due to malnutrition, while the children would develop malnutrition, and at the same time they would get big bellies, and again, their arms would look like sticks basically, and that's what's called that's a word is of African origin perhaps. It's called kwashiorkor, and the another illness is called marasmus, where the kids would be extremely thin with just a skinny belly. And these babies responded differently to microbes because they had different microbial. So what the people who were doing research was they took the poop from one type of thing and transplanted it to others. And their response to the treatment was dramatically different. So again, showing the association of microbial flora to these two types of microbial illnesses. So some responded to feeding with nutritional diet, like high-rich, protein-rich diet, while others did not. So the ones with marasmus responded, while the ones with kwashiorkor did not respond to good feeding habits. And so that has a lot to do, again, the role of microbial flora played a big role in those two types of malnutritional illnesses. Okay, and this is again the chart of how, this is pretty self-explanatory, the chart of obesity versus antibiotic use. And this was almost like an eye-opening chart as to see how where there was more antibiotic use, there was more obesity. Again, and now this may not be the only factor, but at least it makes a point as to how changing the microbial flora may have played some role in leading to obesity epidemic. Okay. All right. And so again, the lean into obesity people were, again, instead of just diet surveys, were more accurately diagnosed based on microbial gene analysis rather than human genetic analysis. Because remember, as we talked about in the first lecture, your own genome is only a relatively small percentage, but the microbial genome is much more diverse and vast in terms of you have only 20,000 genes, while microbes have hundreds of thousands of genes that are in your body. Okay, now these are some rather busy slides as to how there are the lean mice and the fat mice that how they were able to extract energy from the same amount of food, as to see how they give the same diet to lean mice and small mice, as to how they're able to extract different energy. As you can see from the slide on the right, as to the amount of energy that the fat mouse was able to extract was a lot more than the one that the lean mouse. So again, it shows as to how, and this may have simply to do with their microbial flora type of thing. And people transplanted the microbial flora from one to the other, causing change in the behavior. Fecal matter transplant from the fat to the lean, as to, you can see the cartoon is pretty self-explanatory. When they transplanted the fecal matter from the fat one to the lean one, became fat. By the lean, stayed the same, basically. So these are, the middle one are so-called germ-free mice type of thing. It's the center picture. This has a so-called, these are the specially bred mice, the germ-free mice. And when one is transplanted from one to the middle one, became fat, well, no change. So this was, a good proof of concept as to how the blood flora had a huge impact on changing the weight of the animals. Okay, and we already talked about the kwashiorkor, and can be induced in, in mice with the transfer of the microbial flora. And again, the imbalance was between the so-called two microbials that we talked about, called firmicutes and bacteroides. Okay, and again, this explains the same example, that the ratio between the two microbes, firmicutes and bacteroides, became more efficient at extracting energy. And we talked about as to how the food is digested by so-called small chain fatty acids. Okay, and the conventional example of the well-known diabetic drug metformin may again work through this microbes type of thing. So that may have a role in how the metformin works on your diet type of, on your diabetes. Okay. And so the obese people have more firmicutes and fewer bacteroides. Okay, and the microbes from the lean donors, and this is again the animal experiment, caused the weight gain of 27% versus 47%. Okay, so it made a huge difference as to where the samples were taken. Okay, and we talked about akkermansia earlier type of thing. Okay, and this were the twin studies that were done. Okay, and this was an interesting example as to the twins were followed, for a number of years, and again, these were, some of them were identical twins, and the diet had a huge impact, basically. So that was the point that they were trying to make as to weight gain and change that took place. Okay. All right, and let's see. I think I'm going to skip through some of these stuff. And yes, one important thing people had talked about as to how the microbial, some of the foods that were important were, which had microbial foods that were important, but something called, the yogurt is well-known example. And more recently, kefir has become more popular, okay, in terms of microbial generated rich food, okay, and the kefir grain and kefir drinks are the biggest thing that have played a role in changing microbial flora. If you go more ambitious, there are more food called kombucha, and it's a fermented tea that's the next big in thing. And now, last but not the least, I think one food that is the edamame, that is, it's a fermented food, and that again, I had a slide when I did a class on history of smell, and it's called netto, which is the Japanese foul stinking fermented soybean curd type of food, but that just smells so bad, it's an acquired taste. So I think but that may not be as easily palatable. No, it almost reminds you of Limburger cheese type of thing. So the smell was really awful type of thing. And it's only, and some of the Asian cultures are used to eating netto, Netto, that's pronounced. Okay. But again, some of these microbe-rich foods are coming into vogue. As to how beneficial they will be in the long term, that time will tell. And the important part, again, as we talked about in the beginning, was the fiber diversity. And the role of, again, it plays a role in diet and intake in terms of diabetes, as to how okay, now the germ-free mice, and again, the fecal implant, their triglycerides increase after the fecal implant, by extracting more fluid by 2/3, and their insulin resistance increased as well. And it turns out that the microbial flora was a lot more sensitive than the so-called glycemic index that is the vogue for control of diabetes. So, and some people, for example, and it was so sensitive that some people's sugar was better controlled by eating ice cream than by eating rice. And that was almost a flooring type of thing. The people, a guy named Dan Knight, who has done a lot of research on microbial flora, and I have a slide from him later on coming down. It shows as to how microbial flora, so it was individualized diet based on your microbial flora type of thing. So, and so a microbial flora and the small chain fatty acids produced by the gut microbes were a better predictor of insulin response. And again, this may have a role with your genetic profile as well. But again, it showed that this was a lot more accurate way in terms of assessing how you're going to respond to your treatment. Okay. And it's the same way, the bariatric surgery, which is being recommended for people with uncontrolled metabolic syndrome, and it again restored your microbial diversity. So people have done data analysis after bariatric surgery that they were able to restore their microbial flora, basically. Now, again, a couple of the microbes have definitely been established to show a role in colon cancer, especially a lesser known bacteria called Fusobacterium nucleatum was in high quantity in people with colon cancer. And so were Bacteroides and E. coli. So again, and there was definitely a role to be played of this microbe in colon cancer. And there's various carcinogens that were involved and generated by the microbes. And along with the diet that is rich in meat and fat, the nitrates in your meat, it cause the elevation of your in the DNA damage to your colonic cells. And okay, and now, and people have tried treating that with the microbial flora with phage therapy, and some of them have done experiment even with the simple things such as clover, okay, in terms of changing the microbial flora of some of these people. And again, treating microbial flora is not a new idea. It was tried back in the 1890s by something called Coley's toxin. And no, there was one way of trying to treat malignancies. And so, and again, well, we know that the, sorry, excuse me, let me go back. And BCG was used for treatment of bladder cancer. So again, you know, the microbial flora had a huge impact in treating varieties of malignancies. Now, and the response of microbial flora is, again, when we treat certain cancers, for example, melanoma, was a less diversity had impact to immunotherapy when we, the new thing in treating melanoma is called anti-PD-1 therapy, and which is the therapy that Jimmy Carter had. Now, people were less responsive, you know, to immunotherapy if they had less diversity in your microbial flora. And by giving probiotics, again, they became, their response improved. Okay. And now, when we talked about in the first slide about so-called synthetic biology, and the people who are doing research in, it is getting to the point where so-called bespoke bacterial flora, which will secrete enzymes, which will stick to the, I'm sorry, too sensitive of my own. Okay, to the cancer cells. Okay, so that's the part of the research that is being done, is to not only just to genetically engineer microbial flora, so they would secrete specific enzymes and chemicals that would affect the cancer cells. Okay. Okay. Microbial flora has a role also in causing lung disease. It affects the so-called gut-lung axis with the interference with the so-called interleukin 22. It changes the resistance in your lung tissue. And this is, again, obviously, a lot of that is animal work that is done in neonatal lung. As you can see the picture on the right, the neonatal lung, okay, it breaks the receptors and breaks the communication in the immune cells, the so-called CD cells and interleukins. So again, so the lung immunity has a huge role as well. And then obviously the flora that is inherent in the lung is much smaller than in your mouth, and it's harder to culture. But again, you know, this is disrupted by cigarette smoking. Okay, and again, neonatal gut microbiota induces lung immunity against pneumonia, and the role that's played by the maternal antibiotic exposure is, again, has a role in leading to pneumonia. And this is, again, a lot of that is animal research that is done, okay, leading to a propensity to pneumonia. Bacteria and heart disease, okay. Now, let's see. Now, the meat, the so-called choline and carnitine that is in the meat leads to developments of something called TMO that is converted, and it's useful for breakdown of your cholesterol. And again, these are the so-called toxins that take place, and it slows down the breakdown of your native cholesterol, okay, and leads to atherosclerosis, okay. Choline and carnitine are nutrients in our diet, and they change in our body into TMO and TMA and TMO, and again, it increases the risk. Now, an enzyme called FMO3, which can be blocked by drugs, and okay, and that's an alternative. So that's a bacterial enzymes may have a role to play in slowing down the side effects that could take place. Okay. Okay. So I think I'm going to skip that. I already gone over that to some extent. Okay, now, again, the simple drug called the digoxin that's been around for heart disease, that is affected, the metabolism of digoxin can be affected by a bacteria in your gut called Agarnella lenta. So that's one of the times it makes it difficult to get digoxin levels. Okay, so the interplay of your therapy, as you can see how the digoxin is broken down, and if you have this particular bacteria, it breaks down your digoxin metabolite and rendering it ineffective. So that's one of the way it becomes difficult to get adequate digoxin levels. So even the simple drug treatment where we think has an interplay, the same role is there for simple drugs called Tylenol and some of the chemotherapeutic drugs. So the timing of the treatment is interplayed by a variety of your gut microbes. And the digoxin was the prima facie example of it. Okay. Now again, moving further down into the body, the vaginal microbiome is changes. At the time of birth, the vaginal area is relatively sterile, but is colonized by lactobacillus, and it changes with puberty to different thing, and again further change with your reproductive age. So depending upon the age of the woman, the microbial flora of vaginal era changes dramatically, affected again by the interplay, and obviously has a lot to do with the pH of the vaginal fluid also. Okay, normally it is acidic, and some species are more dormant and than others. And even there is even some racial change also, that some of the African American and the Hispanic population have a slightly higher pH type of thing. And again, that is one of the reasons why douching the vagina is not recommended type of thing, because it changes the flora dramatically type of thing. Now, again, if you remember as to the toxic shock syndrome that took place, and due to the tampon that was marketed back in the 80s, the brand name was called Rely, R-E-L-Y, and that changed the vaginal flora and to less favorable and caused the staph infection. The increased absorbency that took place that led to the, okay, excuse me, I think, I'm sorry, we kind of, okay, all right, I think we're out. Sorry, Joan, I think I didn't look at my wristwatch, we forgot the break time, so, okay. Should we break for a few minutes? Because I think I just have a few more slides to do, so we may end earlier than we expected type of thing. So I'll only have about.
Is everyone okay with just waiting and ending early?
Yeah, so I think, yeah, because I may be able to talk about, show you some of my other these things, but so I think, yeah, we should just probably just continue, because I removed my wall clock, so I think I forgot the time type of thing. My apologies. Okay, so this is, I'll just continue with this thing. So I just have a few more materials to cover, basically. Okay, so, and this is the analysis of the human vaginal flora, which primarily it is lactobacillus. Okay, and again, so when you get and this is the 16 as the classical way of classifying the microbes. Here you can see the majority of the organisms are lactobacillus type of thing, which is, and again, it's a lot when. And the common way when you get infection is called, the common process that called vaginal sepsis, called Gardnerella. That is the bacteria that causes the vaginal infection type of thing. Okay, and again, it can change the odor of the vaginal secretions, and again changes with the changes of the pH. I think. And back in the 30s and 40s, Lysol was being promoted for maintaining good vaginal hygiene type of thing, and there were some awful commercials that were being done to promote the use of Lysol type of thing. Again, so it is being discouraged now type of thing. And so actually in tongue-in-cheek, and some of the more modern research is that vagina is like a self-cleaning oven, basically. So it's not recommended to use these chemicals to maintain good hygiene, so to speak. So these are the conclusions that were here. So type and abundance it varies over a short time, and in others it does not change. And it, again, the menstrual cycle has a lot to do with it, type of thing. And okay, and so lactobacillus are personalized for a long time, basically. Okay, and yeah, I don't know what I probably would have to say. We'd have to wait for a long time for vaginal probiotics. Okay. Okay, and even having a different sexual partners can affect your vaginal flora. Now, the more important research that we need to mention before we end is the role of intestinal and brain microbial tissue type thing. And some of the diseases that have been suspected to be related to microbial flora are the classic example being Parkinson's disease. The data is a lot more solid for Parkinson's, and there is some research that I'll talk about for Alzheimer's. And schizophrenia is more observational studies. And depression, again, you can see the role would be due to serotonin and tyrosine metabolism we talked about. There is a lot more on a solid grounds as to the talk between your gut microbe and your brain that takes place. Okay. And the environmental risk factors that are pretty self-explanatory type of thing, as your diet and stress antibiotics have a lot to do with it. And how does the talk take about, which is an interesting feature that we'll dwell into in the next few slides. Okay. And it was interesting that patients who have had, in the old days, before the therapy with newer drugs for ulcer disease, for acid peptide disease, the surgery that was recommended was called pyloroplasty with vagotomy, means actually cutting the vagus nerve that goes to your stomach. And it was observational study was that when that surgery was done, people had less risk for Parkinson's disease. So that was an observational study. And so that gets to the idea as to how your nerves that are related to your brain may have a cross-talk with your intestinal microbial flora. So that led to the starting of thinking as to how the role of your stomach microbes have to do with your talking to your brain. And this is the Dan White, who is one of the pioneer researchers in microbial flora, and he has a big project that called American Gut that we'll talk about towards the end of the talk.
Your gut microbes and your brain communicate back and forth throughout your life through a number of different pathways, and so there are several of these channels by which they talk to each other. So how do gut microbes get their messages to the brain? Well, there are several ways they do this. One way is that immune cells in the gut can sense bacteria in the lumen and release immune signaling molecules, also known as cytokines. Changes in the levels of these cytokines can lead to changes in brain function. Also, gut microbes can activate cells in the intestinal walls that produce peptides. So peptides are short amino acid chains, and these peptides can actually activate nerve endings. Finally, microbes can send direct messages by producing neurotransmitters, in other words, the signaling molecules that your nervous system uses, or their precursors as products of their metabolism. In other words, microbes can make chemicals that allow them to speak the brain's language. Fascinatingly, we think, so one fascinating example of this is serotonin, which you might have heard of, for example, in the context of SSRIs, like Prozac and other drugs for treating depression. It turns out that 90% of your serotonin in your body is in your gut, not in your brain. We also know that the brain and central nervous system have several different ways to communicate with microbes in your gut. Under stressful conditions, the brain will stimulate the release of corticosteroids, which affect immune cells, including the immune cells in the gut. And these can lead to changes in permeability of the gut to microbial cells. In other words, if you're stressed, the microbes in your gut can actually leak out into your bloodstream and have effects all throughout your body. And not only that, these kinds of effects can also ultimately lead to changes in which microbes are in your gut. Also, neurotransmitters have been shown to directly influence gut microbial communities. So next we'll talk a little about behavioral changes. Several different studies have shown that gut microbes can even influence the behavior of the host. We've already mentioned two of these in animals. The first of these is that gnotobiotic mice that were transplanted with microbes from an obese mouse themselves became obese and exhibited the behavior of eating more than they did prior to receiving those microbes and compared to their lean counterparts. What was fascinating about this is that we could cure them of their obesity either by administering antibiotics or by cutting off their food supply, so just putting in their cage the same amount of food that a normal mouse would eat. Another really interesting study showed that when aggressive mice were given antibiotics or probiotics to change their gut microbes, they became less aggressive. And in case you're thinking that these things only apply to mice, here's another experiment in fruit flies. And this experiment showed that flies that were raised eating sweet food, in other words, molasses, preferred to mate only with other sweet eaters, as opposed to those who were raised on a starch-based food that was considerably less sweet. It turned out that the same was true in reverse. In other words, the starch eaters preferred to mate with starch eaters. This difference in mating preference was tracked down to the presence of bacteria in both the food source and the flies. So maybe you can't catch more flies with molasses, but if you're a fly, it can help you decide who to mate with. Another example we'll discuss is autism, which is still very controversial, but the preliminary evidence to date suggests that studies of children on the autism spectrum may have different microbes in their gut than neurotypical children without ASD, often their brothers and sisters. Now, while there is a relationship between these gut microbes and ASD, the causality of this relationship is unclear. And in part, the reason for this is that autism often co-occurs with diarrhea and other gut problems, which might themselves be changing the gut microbiome. As a result, it's really difficult to find the controls that you need of children who have the kinds of gut barrier dysfunction that you see in autistic children, but who themselves are neurotypical. So we have to be very careful before drawing any causal or correlative links in this case, until we have additional information with more careful controls. However, we can do a lot more in mice than we can do in children. And one fascinating study conducted by Sarkis Mazmanian's group at Caltech involved activating a pregnant female mouse's immune system by simulating a viral attack using double-stranded RNA. Now, when the mothers are under this maternal immune activation scenario, basically a model for RNA viruses, like for example the flu virus, what Sarkis saw was that it led to autism-like symptoms in the offspring. And so, what were these symptoms? Well, they included things like repetitive behavior, lack of communication and social interaction, cognitive impairment, and just like in children, gut barrier dysfunction. Now, along with all of these phenotypic defects, the gut microbial community was also altered after maternal immune activation. However, using a probiotic bacterium found in the healthy human gut, Bacteroides fragilis, could actually partially restore some of these issues. So how did this work exactly? Well, profiling by metabolomics, in other words, reading out all the metabolites that were in the gut of these mice, showed that one particular chemical, called 4-EPS, was at much higher abundance in the maternal immune activation mice, and much lower after restoration with the B. fragilis probiotic. What was really fascinating, though, was that administering that one chemical that had been identified, 4-EPS, to otherwise healthy infant mice could cause some of those same autism-like symptoms. So this study is especially important because it suggests a general pipeline for linking microbes, metabolites, and health, including conditions that affect the brain. The first step is to identify altered community profiles, then to find out what metabolites are involved. And then finally, we can test those microbes and or the metabolites that we think are important for specific effects in preclinical models, and then ultimately in the clinic in humans. The final topic we'll talk about is stress, anxiety, and depression. Many of the interactions we observe between the gut microbiota and the brain involve stress in some form. Mice, chickens, and even primates that are put into stressful situations show substantial changes in their gut microbes. You can see in this figure, produced by Bailey and Co. in 1999, that as infant rhesus macaques show more stress-indicated behaviors, the relative abundance of lactobacilli, often considered to be a beneficial microbe, tend to drop. Another study by Pseudo in 2006 in mice has shown the reverse relationship, where stress actually increases the amount of lactobacillus in mice. And then germ-free mice that are raised without any microbes of their own in a germ-free bubble turn out to have a stronger stress response when they're restrained than do mice with a normal gut microbiota, suggesting that your normal gut microbes might actually increase your resilience to stress, to some kinds of stressful conditions. Moving on to depression. Depression is a major cause of disability in the United States and even in much of the developing world. In fact, the National Institutes of Mental Health estimates that by 2030, depression will be the leading cause of disability-adjusted life years, making it an especially important disease to tackle. We already know that depression is linked to the immune system, and because microbes can influence and regulate immune responses, it makes sense to think that by manipulating the microbiome, we could treat or even prevent depression and its relative stress and anxiety. A fascinating study in mice conducted in part by Chris Lowry showed that the soil bacterium Mycobacterium vaccae can decrease anxiety in mice and can even help them cope better with stress. This could be a model for treating anxiety orders like PTSD in humans. Who knows, one day we might even be able to vaccinate against depression and against PTSD using microbes. Graham Rook, Chuck Rezan, and Chris Lowry proposed exactly this idea after seeing evidence from studies in mice that it just might work. In fact, a study by Masadi and colleagues showed that people taking probiotics with lactobacillus and bifidobacterium, again, two kinds of beneficial bacteria that are especially prevalent in the infant gut and are involved with short-chain fatty acid production, showed substantial reductions in anxiety symptoms. So in conclusion, we're only beginning to understand the connections between the influence of the gut microbes on the brain. However, already there's evidence that beyond diseases, our gut microbes can have an effect on the formation of our memories, our emotions, and even on the way we make decisions. So therefore, just like our brain, our gut microbes are part of what make us human, and we cannot neglect their input into our behavior, our moods, and perhaps even your gut microbes.
Okay, so I think, this was rather long, but I thought, he went through all the points and I was able to rest my voice because I've been talking too fast. Sorry. And so this is, basically how the talk goes about, just to belabor the point. HPA is the hypothalamic pituitary axis, and that's the classical hormonal pathway that you may have studied about in school. And the vagus nerve that we talked about, serotonin, and the GABA is the another brain neurotransmitter. So the three ways the talk goes on between your brain and your body. And this was an interesting thing this just made, it's like how the Alzheimer's is far less common in the developing society, and it's increasing. And so this was again, oral hygiene was related to dementia type of thing, and so brushing decreased part of the inflammation that we talked about earlier type of thing. But the more interesting thing is probably this one type of thing as to how Parkinson's disease has been related to dysbiosis. And I know it's well known clinically that people with Parkinson's have a lot more constipation type of thing. And so there is distinct, more solid ground that we are on when it comes to Parkinson's disease as to how the gut microbes is maybe playing a role, as to signals from the gut microbes are required for the inflammatory response. And there's a classical hallmark protein in Parkinson's disease called synuclein, and that may be related to the dysbiosis that takes place, and now there's a big role. Initially, the cells in your brain called microglia were considered to be just kind of useless. The more important cells were pyramidal cells and the astrocytes and other things. But the role of microglia is coming into vogue. A lot of the neuroscience research focuses a lot more on the microbiome role type of thing as to the dysfunction that takes place between the Parkinson-derived microbiota versus the typical microbiota. So on Parkinson's disease, we are a lot more solid ground as to the role. Okay. And this came out recently in the Science Journal about the herpes virus and the 3 other bacteria which lead to gum disease and had a role with the amyloid, beta amyloid, which is the hallmark of Alzheimer's disease. So this is about as recent a report as I could find in terms of role of various microbial agents and related to Alzheimer's disease. Again, this is a very preliminary work as to how the microbes can affect the microglia in your brain, which are the communicating cells, and the infection can release cytokines, again, leading to, in your neuron, the amyloid precursor. This is the hallmark protein of Alzheimer's disease, and leading to plaque formation. And this just was dramatic finding that I just came across within last, I should say within last 6 months type of thing. So that's why I labeled it hot off the press. So interplay between infection, immunity, and amyloid. And there are varieties of research that is done with, like there's a project that Dan White, whose video that we listened to, is pioneering in something called crowdfunded US through University of San Diego, UC California, San Diego, where you donate $99 and you can send your poop and they'll send you the analysis of your microbiome. And there are varieties of other commercial firms that are also doing that. Now I checked out on website, and due to COVID, they have stopped doing this right now. But that'd be one way to get some information and also to contribute to research. They're using the data for increasing their information or database, so to speak. It's a project called American Gut. Now, we talked earlier about the drugs that we talked about. This was a slide that I should have moved it further. The drug response, the varieties of drugs are affected. We talked about digoxin, the various chemotherapeutic agents, the PD-1 inhibitors that are affected by your microbiome, lipids that we talked about with the bile acid. So, and again, so that this was Dr. Lynch's data. The medications, as many as 1,200 micro drugs may have some role in with interplay with your microbiome. Okay, and we talked about earlier with rheumatoid arthritis, that was proposed as far back as almost 100 years ago, the role. Okay, all right. And it was interesting, the study that was done for lipid, that people who were treated with a well-known drug called Crestor, some responded dramatically, while other half, about 45% responded dramatically by dropping their LDL, but as many as 40% didn't have that dramatic a response, while 11% had no response to the more potent drug called rosuvastatin, brand name is Crestor. And even actually, and that just ties in with the role of bile acids that we talked about earlier, as to how, so when you fail to see a response to a drug, so it was related to the role of bile acids and your microbial flora. Last but not the least, this was almost blew my mind as to, I think that even some of you may have read a book called Body Farm that was written by how the maggots grow on your dead body type of thing. And the classical example that was done was, which depending upon the timing as to when the insects settle on the dead body type of thing. But not only that, the science has changed, as to how not just relying on the flies and the maggots that you can see, but also the microbial flora has a role in determining in forensic pathology. That people who are doing some cutting edge research and not just relying on the bugs and the insects that you can see on the dead body, but also the microbial flora that may have a role in playing forensic pathology, that type of thing. So So again, so the science has increased, not just from what you can see to what you cannot see. Okay. Now again, no, this is called probiotics and prebiotics. These are the probiotics, okay, and prebiotics, which would help balance intestinal flora, and so it's a big business right now, type of thing. Okay, and well, there's something called prebiotics, which indirectly stimulate the bacterial species, type of thing, and we can modulate inflammatory function. So this is the two big categories in which the commercial agents are available. Now again, the important part is not to jump the gun on this, because these are the ones that are manufactured and are sold in stores are the ones that grow more easily, on industrial uses. But again, these are not all that they're cracked up to be, because when you take them by mouth, a lot of them actually just get washed away. And the analogy that is given is to like having something set with with two open windows on both sides, so they just go right through you basically. So it is not sure as to how much benefit actually you're getting, type of thing. And because the manufacturers are using going for the so-called low-hanging fruit, the ones that they can grow more easily in huge vats and in chemical in the industrial plants. So and these were some of the data from the American Gut Project that Dr. Knight talked about, type of thing, that they are collecting samples based on volunteers who have actually contributed to people-based science, type of thing. And the microbial flora changes as we get older and becomes, and the antibiotic use, as we already emphasized, and the plant-based diet has a lot more diversity that we talked about. And relative alcohol consumption also affects your microbial density. And this is interesting. This may give you an incentive to have some drinks as to have more diverse microbial flora, that thing. So, and the food is the language that speaks to our genes, so this is an important thing. So with so-called anti-inflammatory diet, they're going back to what is well known as the so-called Mediterranean diet. It's the more diversity is important. It's not what you eat, but how much diverse that is. It's like including varieties of fruits and vegetables at the bottom part, including a lesser amount of meat, and increasing. So this boils down to what was known in cardiology as the so-called Mediterranean diet, is more diversity, type of thing that was emphasized. Okay, and again, this beleaguers the point as to the role of Mediterranean diet, more plant-based food, and giving more diversity, I think. So again, it's a pretty self-explanatory that we already elucidated. Okay, and this again emphasizes what are probiotics. The primary one that is being marketed is called lactobacillus and yeast. Those are the two workhorses that have been known from times immemorial with formation of yogurt and acids. And these are the ones that are being promoted in. And this is just an example as to how much this is available at your local pharmacy. You can see this is what's being sold on the drugstore with varieties of claims, type of thing. Some of them are legit, some of them are kind of over the top type of thing. So, okay. Now we do need microbes, but we're not quite sure as to how good will they do, type of thing. So and these are some of the references that we talked about. This is the book by he has done a lot of work on H. pylori. I already showed about that's on my shelf, if you can see behind me. I talked about Dr. Finley's book about Let Them Eat Dirt. It's a very easy to read book. Some of these are, this is especially the mind-gut reaction. This is a hard read about, he's I think he's European, and it's a mind-gut reaction. It was a more difficult read, but the first three ones I would strongly recommend for those. And Missing Microbe by Dr. Blazer, who's been a pioneer researcher in H. pylori, and Ed Young, who talks more about not just human body, but also the microbes and the ecology of microbes that we talked about in the first half about symbiosis. A lot of the data is borrowed from him. And this is the person who actually was the pioneer in microbial research.
We live
Dr.
in a
Jeff,
microbial world, a world dominated by
he was interviewed on 60 Minutes, so I think this may be as a part of that.
microbes. My name is Jeff Gordon. I'm a professor at Washington University in St. Louis, and we studied our microbial cells, the microbes that live in our bodies. We humans are no exception. There are more microbial cellular parts to a body, more microbial genetic parts to our genetic legacy. And we're very interested in understanding those interactions between parts. What aspects of these interactions determine our health, our disease risks, and our differences from one another. We were interested in this amazing organ called the gut that's 10 meters long, where different functions are portrayed or expressed along the length of the gut. Our microbial communities are a factor that affects risk for obesity and mediate not only the amount of fat that we store, but also the type of metabolism that we exhibit. Food is incredibly important, and that in the future, if we're to look ahead, we can imagine installing microbes that are missing that can prevent or treat obesity, but doing so in a proper diet context. Prevent or treat in the context of the microbes of the population that we were interested in, because we could sample microbial communities from people living in Saudi Arabia, living in Malawi, anywhere in the world with appropriate approval, and take the culinary traditions of that society and add those traditions or apply them to mice. I do believe that all the things that we have done here together in this particular set of explorations scientifically allows us just to begin to do what we've always dreamed of doing, and that the best part is in front of us.
Okay, so that was my last slide, and I think on that end, we'll just say goodbye to all of you, and I'll stick around for questions, comments, suggestions.
Murad, I want to thank you for all the work you've done on this. This
Okay,
last
always a
part
pleasure.